and the organized committee members, especially Dr. Murase, for inviting me to this symposium. I'd like to begin my talk with this question. Why do organisms have the lifespan? In prokaryotes, there's a big group of bacteria. Asexual reproduction can repeat asexual reproduction indefinitely, so that they are considered to have no lifespan. But in eukaryotes, the continuity of the asexual reproduction is intervened regularly by sexual reproduction. So this duration, being with sexual reproduction, is the lifespan. In asexual reproduction, 100% of cell is conserved, while in sexual reproduction, only 50% cell is conserved. So the question is, why this sexual reproduction can intervene in the asexual reproduction process? The eukaryotic life strategy can be viewed like this: setting the limit on asexual reproduction in soma, that is a lifespan, in exchange for the survival of germ through sexual reproduction. Then the question is why this exchange occurred? Because sexual reproduction results in genetic diversification. This is a general story. Now, as a model organism of a eukaryote, I'd like to introduce my lifetime friend, Messium. This is a shaded protozoan with whom I have studied the lifespan for a long time. And the question is, do Paramecia have the lifespan? This question was a very big problem in the beginning of the 20th century. In this question, the lifespan refers to the limit of cell division. So the question can be asked whether the rotation of cell cycles can continue definitely or indefinitely. Okay. So this question was first challenged by Woodruff of Yale University. He maintained stock of Paramecium by Aurelia without allowing sexual reproduction for 33 years, from 1907 to 1940. And he finally concluded that Paramecium by Aurelia is immortal. But this story was denied by Sonemon of Indiana University in 1954. And he found that this species undergoes a special type of sexual reproduction called autogamy. So he overlooked autogamy, and Solomon concluded that these species are mortal. This change from immortal to mortal also occurred in the other species of Paramecium caudatum. The Russian scientists, Galaziev and Metalniko, studied in the same way for 22 years, and also concluded that this species is also immortal. But this was denied by us in the 80s. They overlooked cell-gene conjugation, and we demonstrated that this species also has a definite potential of cell division. Okay. Then, why do Paramecia have the lifespan? This eukaryotic strategy that is setting the limit on asexual reproduction in soma, the lifespan, in exchange for the survival germs through sexual reproduction. That's true for Paramecium. But how about sexual reproduction results in genomic diversification? Can this be the reason? No. So, why do Paramecia have the lifespan? This has long been my puzzling question. Today's talk is an answer to this question. First of all, I'd like to introduce the psychological process of autonomy in a more, I'd like to have a closer look. Paramecium tetraurelia have two different nuclei. One is somatic macronucleus, and the other one is germ micronuclei. There are two micronuclei. So during autogamy, two diploid micronuclei undergoes meiosis to result in 8 haploid nuclei. Then 7 of them disintegrate, and the remaining one undergoes an additional nuclear division to result in a gametic nuclei. So they fuse to produce fertilized nucleus. Then they differentiate into soma and germ. During this process, the old macronucleus disintegrate and finally become extinct. Because of this cytological process, all of the genetic loci become homozygous, you see. So once autonomy occurs, then in the next generation, the genotype remains unchanging, even after recombination occurs. So as you see here, this asexual reproduction called autonomy, this occurs only in a single cell, irrelevant to the sexuality, and no genetic diversification occurred. But still, autonomy is considered as sexual reproduction. The reason is given here. In mammals, gametes such as egg and sperm produced by meiosis from germ undergoes fertilization. Then fertilized egg differentiate to soma and germ. And also in seeds, including Paramecium, gametic nuclei produced by meiosis from germ undergoes fertilization, and the fertilized nucleus differentiate to soma and germ. This occurred in a nuclear level, not in the cell level. But you call this process sexual reproduction. Then why not this process? And actually, once autonomy occurs, the single cell finished this process undergoes asexual divisions, passing through autonomy maturity, and senescence. And finally, it dies with clonal death. And this process is counted by the number of fissions or cell division. For example, Paramecium tetraurelia has a lifespan of 200 to 300 fissions, and P. caudatum has 600 to 700 fissions. And during this maturity, if cells are put into the food depleted condition, then autonomy is induced. Then again, food is given to them, then they come back to the beginning point, and then starting the next generation. So by this process, time is reset to 0. Okay. So I repeat again. If this, once you recognize that you take this process as sexual reproduction, then the fundamental events that characterize sexual reproduction are not sexuality, not genetic diversification, but rejuvenation, setting the time, developmental time to 0, and meiosis and fertilization. This process should be regarded as essential. Then, autonomy information suggests that 1. Autonomy is a form of sexual reproduction that does not result in genetic diversification. 2. Autonomy is not a complete form of sexual reproduction. 3. A primitive form of sexual reproduction should exist before the complete form of sexual reproduction emerges. The primitive form of sexual reproduction should be related to the emergence of the eukaryote. Then the next question is how eukaryotes, first eukaryotes emerge. Eukaryote emerged about 2 billion years ago. There is no generally accepted theory about how the first eukaryote emerged. Some people believe that end symbiosis can explain it, but I don't think so. It's not likely, because for any end symbiosis hypothesis, end cytosis of symbiont by the host cell is essential. It's mandatory. But that power, that ability of end cytosis is just function characteristic specific for the eukaryote. Okay? So this theory premises the existence of the eukaryote for the explanation of the emergence of eukaryote. That's the reason. I propose genomic and cellular enlargement is a very essential primary instance to give the way to the first eukaryote. Genomic enlargement make it possible that many new proteins become available. And among them, actin and tubulin are most important. They are used to make cytoskeletons. Cytoskeletons make the end cytosis possible. And cytoskeletons, of course, supported the big cell. And cytoskeleton also produce the internal membrane system. And this internal membrane system was very useful to compensate the big cell. First of all, this is prokaryote. 1 micron of cell become increased in diameter tenfold, and it results in 100-fold increase in the cell surface area and 1,000-fold increase in cell volume. Okay. Now, inside the internal membrane also make it possible to create the nuclear membranes. But there is this enlarged genome brought a new problem. Enlarged genome requires elongated time for each duplication. Elongated time for duplication increases the frequency of harmful mutations. Safety measures for enlarged genome and cell are needed. I like to propose that 2 safety measures emerge. One is differentiation of germ and soma, and the second is a change from haploid to diploid, from end to end. These safety measures first appear in the being eukaryote. Here is a summary of my original hypothesis, proposing that lifespan and primitive sexual reproduction were the consequences of the safety measures. Differentiation and German soma resulted in prolonged lifespan. A change from haploid to diploid resulting in air-accumulated lifespan on one side and primitive sexual reproduction on the other side. Today I'd like to talk exclusively of this process shown by the yellow line. So my idea was derived from the consideration with respect to essentials of sexual reproduction shown by autonomy. As shown here, as I said before, sexual differentiation and genetic diversification are not seen in autonomy, but rejuvenation, reforming soma from germ, and meiosis and fertilization are seen in autonomy. But of course, none of them are seen in asexual reproduction. But if you take meiosis and fertilization as haploidization and diploidization, the changes from 2M to N, and fertilization as M to 2M, then these processes are common in both asexual and sexual reproductions. So it means primitive sexual reproduction may look asexual. Sexual reproduction looks asexual. Asexual reproduction of the haploid cell is repeat order duplication and distribution. In other words, diploidization and haploidization. The diploidization can be caused also by cell fusion, and if it followed by distribution, then it is no less a process than one-step meiosis. The asexual reproduction of the haploid cell is transformed to the asexual reproduction of the diploid cell, but eukaryotic cells with an enlarged genome have to change from N to 2N for safety. This is a safety measure. So diploidy is a safety measure because loss of function induced by mutation of a gene is covered by a complementary gene. So this process of the diploid cell cycle is now shown by the capital. But this process cannot be, the DI module of the diploid cell division cannot repeat infinitely because the function of diploidy to cover the harmful recessive mutation would someday become not to work anymore. So it is necessary to change to the haploid. Haploidization before the accumulation of mutation becomes serious will result in rejuvenation. So I like call this process is no less a process than asexual reproduction alternating between haploid and diploid. But I named this process primitive sexual reproduction because the rejuvenation, diploidization, and haploidization, those are the minimum conditions to be called sexual reproduction. Okay. This process is important because also genetic diversification to some extent can be expected through the evaluation of usefulness of accumulated mutation. So this means accumulated mutations are expressed in the haploid cell, and some combined mutations could be advantageous. Otherwise, disadvantageous if worked individually. So this process makes some kind of diversity. And I would like to stress that this process can accept also the diploid produced by cell fusion. If cell fusion occurred here and followed DI, this is just one-step meiosis. Okay, and this can also be acceptable for the two-step meiosis. If this diploid cells was followed by DU-DI in this way, this is just this can be easily acceptable for the two-step meiosis. And the timing of sexual maturation is also become very meaningful, because this timing, 2M to N, this change is just similar to the age of sexual maturation. So this new idea of primitive sexual reproduction will give an answer to some puzzling questions. One of the examples will be shown in the last slide. So this process from N, the life cycle tactics, from N to half to N and N and almost to N. This is the way of the evolution, evolution. But it is generally believed that some protozoans, such as amoeba and euglena, have this type of cell cycle, life cycle. But it appears unrealistic, because this has already been a eukaryote and have the bigger enlarged genome. So I thought this cell cycle have two facets. One is for these groups. This is consisting of fertilization, the germline fertilization and germline meiosis. But for amoeba and euglena, these are undergoing a similar process, but not fertilization, meiosis, but only the diploidization and haploidization. This is just a process I call primitive sexual reproduction. Thank you.